Well, I want to express my gratitude to Encarnita and Victor for the invitation to be here. First time at the AIRRC meeting, and really impressed with the level of attention to detail and rigor. I have to admit, I have a little bit of rigor envy because in the field that I work in, which is sort of adjacent to repertoire sequencing, there's really a lack of standards. And so my goal today is to hopefully convince you that the adjacent field of antibody reactivity profiling is a worthwhile activity, and also to recruit your help in establishing the rigor in that field that you've done in yours. So I think we're all here because we appreciate the breadth of the central importance of immune responses in many health and disease states. So really immune responses are shaped by your inherited genetics, along with your lifetime of environmental exposures, the random stochastic mechanisms that the immune system uses to generate diversity. And all of these contribute to our health, and either indirectly or directly can be associated with disease states. So I like to think about antibodies as a way that the immune system can tell us its story. We have highly complex repertoires of these protein molecules in our blood, and actually the abundance of the molecules is quite staggering. So there's about 800 trillion antibody molecules per 20 microliter drop of blood. And so if you imagine an antibody molecule being a word, you can ask how many novels could be written in a drop of blood, and actually there's one for every human on Earth. About 8 billion novels could be written in antibody molecules. And this repertoire is sculpted over our life by all of our exposures, microbiome, infections, genetics, vaccinations, and also by our demographics, age, nutritional status, etc., and even the presence of neoplasms that the immune system is seeking to recognize. And the repertoire, in addition to providing humoral immunity, is really a deep source of information that we can mine in the form of biomarkers. And it's really useful in the sense that it maintains a memory of prior responses, so even after the antigens are gone, they bind their targets with high affinity and are really stable to freeze thaws and storage. So it's an ideal analyte in a sense. The constant region of the antibody, the isotype, can reflect the context in which the immune response happened. And it's really easily accessible and in high abundance. So I'm going to entertain you with an idea about where the medical field is going, and give you a scenario of me visiting my doctor in 10 years from now, getting my annual checkup. And of course, this doctor is going to be an AI chatbot avatar that accesses all known medical knowledge. And because of other science advances, I now have a full head of hair. So he wants to talk about my antibody profile, my antibody reactome, and how it's changed over the last year. In all of these examples, we actually have data from my lab and others to support the feasibility of these types of reports. So he's letting me know that he found a cancer reactivity, but don't worry, because it was apparently a productive response, and so most likely cleared the tumor, but out of an abundance of caution, we're going to do some body scanning to confirm that there's no tumor there. Did a profile of the antiviral antibodies and found that my titers were good, so can skip the vaccine this year. That's great. They know I filled out a questionnaire and mentioned that I'm going to move for a new job, and they did a profile of my IgE antibodies, and now he's offering to provide a geographic map of compatibility with my allergy exposure profile. And then he mentions that they detected a new lupus autoantibody with a 5 to 10 year prediction horizon. And this is really true. These antibodies come up many years before disease expresses itself. And based on existing evidence, it's going to propose an immune reboot where I do some diet modifications, take a short course of oral steroids, going to do another profile, and then if it's still looking like an issue, maybe we'll do B-cell depletion therapy as a preventative measure for an autoimmune disease. Thanks, Doc. Wow, a lot's happened in my antibodies over a year. But really, I want to highlight that all this information is there in the circulating antibody repertoire about cancer, autoimmunity, infections, and susceptibility to infections, and our allergies. So we think a lot here in this community about sequencing, and can we really read these types of stories from cells that may or may not be abundantly available in the periphery? Maybe mass spec direct sequencing of antibodies will really develop over the next 10 years. But even if so, can we predict their targets if we know their sequence? And I think what's more likely, and what's already been developing, is an indirect measurement of what they bind to. And so, I think we're all familiar with ELISAs, measuring one antigen specificity at a time. But really what we need are technologies that can provide the equivalent of hundreds of thousands of ELISAs simultaneously. And this is what we refer to as antibody reactome profiling. You may be familiar with some of these platforms that have been developed over the years. For instance, microarrays, protein microarrays, or peptide microarrays, really just a highly multiplexed ELISA. They typically use full-length antigens, which is a great benefit, but have been challenging to standardize based on the quality of the antigens, the fact that they're dried on a surface, throughput on a slide is pretty low, and they tend to have very high costs, limiting their use. So an alternative that's lower cost and provides better antigen quality control are bead arrays like Luminex, but these are pretty limited in the level of multiplexing. With the development, the advances of high throughput DNA synthesis and sequencing, we and others have adopted and developed molecular display platforms like phage display or yeast display to leverage the massively multiplex nature of DNA synthesis and sequencing. So these can be very highly multiplexed in the hundreds of thousands, but tend to only display short to peptides to protein fragment lengths on their surface. So over the past couple of years, we've developed a technique to overcome some of these limitations called molecular indexing of proteins by self-assembly, or MIPSA. And basically the goal is just to put a simple DNA barcode directly covalently attached to antigens in a high throughput way. And this has the advantage that both peptide libraries encoded by oligonucleotide libraries and open reading frame libraries can be expressed and presented as an antigen library at the same time. And the limitation at this stage is that these libraries are created in E. coli cell-free lysates, but we're working on some eukaryotic alternatives. So the chemistry that we're using to link the DNA barcodes to the antigens. It involves the halo tag system, the halo tag is a 30 kilodalton protein that can be fused to a protein or peptide of interest, co-expressed. And it forms a covalent bond with a halo ligand, which is a small molecule that can be covalently attached to DNA barcodes. So this is a great system, but the way it's drawn here, you'd have to attach the DNA barcodes to the antigens one at a time, and so that's not very scalable. So what we developed with MIPSA is a self-assembly process that basically allows the protein to add its own DNA barcode during the translation of the antigen library. And Joel Cradle here is a postdoc in the lab who worked on this and now is the director of R&D at Infinity Bio. So with this technology, we're able to really convert an antibody profiling question into a DNA synthesis and sequencing question. So we can take databases of antigens and use the MIPSA technique to convert those into barcoded antigen libraries. And some of the advantages I'll mention, if we're trying to cover protein sequences that are not previously cloned, we can design peptides that tile across each one of those proteins. So what I'm showing you here is a 60 amino acid peptide tile that is part of the spanning peptides across a protein. And then we'll stitch together the DNA encoding each of these peptides with a halo tag encoding gene and a DNA barcode. And so this is a cell-free assembly process that we use to maximize uniformity, minimize any dropout. And then we'll make RNA from that DNA construct and reverse transcribe it with a halo ligand-associated primer so that we now have a cDNA barcode. And then we can translate this library of molecules and end up with sets of antigens that are either full-length or peptides and their associated barcodes. Once the library is made, they're actually very simple to use. You just mix a blood sample if you're doing serum antibody profiling. We can also use this for monoclonal antibody analysis. Capture those antibodies and the antigens that they're bound to on magnetic beads, wash away all the unbound antigens, amplify the barcodes, use sample indexing and deconvolution to read out the antibody reactome of each sample. And this is a relatively high throughput process that can happen in 96 well plates and involve liquid handling automation. So our sort of proof of concept was looking at COVID-19, severe disease, and coming on the heels of some really nice work out of Jean-Laurent Casanova's lab, where they identified some type 1 neutralizing interferon autoantibodies occurring in a subset of individuals with severe life-threatening COVID-19. So we looked at a Johns Hopkins cohort and performed an unbiased full-length human proteome-wide MIPSA screening campaign and identified those same type 1 interferon neutralizing autoantibodies in a subset of these patients. So this is just 5 patients here. We also saw a couple of individuals that had type 3 interferon neutralizing autoantibodies. And we confirmed that they were actually functional in a cell-based assay with interferon lambda added to the cell culture after pre-incubation with the patient's antibodies. And this study found that it was actually really important to be looking at full-length proteins, which is not uncommon for autoantibodies because they tend to recognize more highly conformational epitopes. So it really underscores the complementarity of antigen libraries that are peptides that can cover everything, along with full-length proteins to capture as much conformational epitope space as possible. So just quickly, some of the benefits of MIPSA are the ability, as I mentioned, to simultaneously detect antibodies targeting full-length and peptide epitopes. There's relatively low non-specific binding because of the very small tag. With our barcode representation system, we have much more robustness in being able to look at many barcodes associated with each antigen. These are all GC balanced barcodes to allow undistorted amplification of those sequences. And we have this system for ensuring that the data that we're looking at is actually only associated with wild type or intended sequences that we encode with the oligonucleotides because oligonucleotide synthesis does have errors. So we're only looking at the barcodes associated with those perfect coding sequences. So because of these advantages, we started a company to enable the research community to access the technology. The company is Infinity Bio, based out of Baltimore. This is a 9,000 square foot lab space before we fill it up with automation equipment. And the team here, we have a booth, please come visit. Infinity Bio is a service business, so we just receive samples and send you back the data. There's no IP restraints or anything like that. We're excited about the catalog of libraries that we currently have available and are also able to work with customers to develop custom content. Please contact us if you'd like to see some performance qualification data. And I wanted to just tell you a little bit about how some of the customers are using the services. And so in the setting of a longitudinal study, you can see the emergence of new reactivities in a totally unbiased way between two time points. Reactivities showing up more strongly at the second time point than the first. Decline in reactivity strength due to waning, indicating a recent response that didn't happen during the interval. And then most of the responses that we detect, though, are very stable over time, and these reflect the historic immune responses. In cross-sectional studies, looking at cohorts with and without a disease phenotype, for example, we can identify exposures that may link to disease, like Epstein-Barr virus and multiple sclerosis, for example. Identify different responses to the same exposure, like different responses to vaccination, leading to different levels of protection, for instance. Autoantibodies and their mechanistic basis, examples like in myasthenia gravis and that target. And then, of course, identify new biomarkers that can provide clinical utility in diagnosis and risk stratification. And thinking about mechanisms, antibody reactome profiling provides an important new key piece of the puzzle in understanding disease mechanisms. For instance, understanding how genetic associations may exert their effect through immune responses, linking the antigen specificity to public antibody sequences, for instance. So as I mentioned, the field of antibody reactome profiling really does need standardization because the question is not how well does any specific technology work, but how well is it going to solve a specific problem? And so to be able to compare across platforms, we're really unable to do that right now. And even unable to make data available in a way that allows people to compare platforms and reproduce studies. So I'm just going to make a pitch for these standards that I imagine need to be implemented, but don't have the experience of this group and actually seeing that to fruition. So I'd love to hear, about the feasibility and steps that might be taken to achieve these goals. So one thing that's very clear is that we need standardized analytes that can be run on the different platforms. So these would be like a collection of monoclonal antibodies that could be library specific. So are they anti-human antibodies or antiviral antibodies to be tested on the different platforms? They should target different types of epitopes, linear, conformational, post-translational modifications, and they could be pre-mixed in different concentrations, low concentrations, high concentrations, and preferably these would be blinded to the users, and the data would be uploaded and analyzed by a third party. There's currently no repository for antibody reactome data. And so, for instance, when my group publishes a paper, editor will often ask us to deposit the data in the appropriate database, and we have to reply that there isn't one. And so I really think that there needs to be this for sure. It needs to be queryable, and there needs to be a minimum amount of data associated with it, like in AIR data, for instance. So it would really need to include information about the platform, sample metadata, obviously the antigen library member metadata, and that should include the sequence and any sort of quality control information associated with that particular antigen member in the library. We should be able to deposit our raw data and then process data in the code that converted the raw data into processed data at a minimum. There's a lot of different ways to use antibody reactome data. I described longitudinal analysis and group-wise comparisons. So these pipelines really need to be transparent and reproducible so that others can use them and reproduce findings. Looking at epitope profiling allows you to look at the clonality of an antibody response and even aggregate over, for instance, peptides tiling across an entire species of virus. Antibodies, as we heard about it, can be very cross-reactive, especially with homologous sequences. And so there's a real need to be able to deconvolute antiviral antibody responses in the setting of viral antibody profiling. Oops. And then, of course, many researchers in this field could benefit from guidance on their study design, how to establish power to detect signal of an expected effect size given prior studies. We need to control type 1 error to avoid publishing false positives, and there's a lot of work that can be done in how to design these antigen libraries to make them most efficient. So I imagine if all this was successful, we could really do a good job in comparing platforms and determining what's the right platform for the right question. I imagine there could be a third party that would generate like a report card that's associated with that set of spike-in antibodies and can report on how comprehensive the library is, how well it detected the antibodies who had targets in the library. Could tell the difference between isotypes. And then how many false positives are generated per set of unrelated antigens, and how reproducible is the assay, how concordant are the signals, and do the signals correlate with the level of the analyte, the antibodies. And I think that's the missing piece from being able to evaluate all these platforms and select the appropriate technology. I'll just leave you with a study that we did in a clinical setting. So just to convince you, hopefully, that it can be useful in a clinical setting for diagnostics. This is unbiased viral antibody profiling from individuals with encephalitis, suspected infectious encephalitis. And I'm just showing you for every library member, there's about 100,000 peptides here, the reactivity level at day 1 versus day 14 when they came into the hospital. So anything off diagonal here is a new reactivity. And so we're able to first identify what are the changed peptide reactivities over that 2-week period in an unbiased way. Identify which peptides were related to each other to determine the response clonality using a network approach. And then using sequence alignment to all the viruses, figure out which were cross-reactive and not, and developed a framework, a statistical framework for deconvoluting those cross-reactive responses. We could report then the evidence for each of these infections. And layering this on top of the standard clinical testing, we're able to improve the diagnostic yield by about 44%. So in summary, circulating antibodies provide a wealth of health-relevant information about cancer, autoimmunity, infections, vaccines, and allergies. Advances in DNA synthesis and sequencing have enabled massively multiplexed antibody reactome profiling technologies, but the field is in dire need of defined analytes, like monoclonal antibodies, and standards for data storage, data sharing, and analytical pipelines to enable this imagined future of antibody profiling with your doctor. So I want to thank all the contributors to these studies over the years and the funding agencies that have supported the work, and thank you all for your attention.
That was great, thank you so much. So we have about 5 minutes for some questions. So starting over here, one side of the protein seems to be covered by halo tag. So these epitopes are lost for the screening?
Yeah, that's a great question. So right now we fuse the halo tag to the N-terminus. And so yes, if the epitope is close to the N-terminus of the protein, the antigen, then it could be sterically hindered. In the future, I think we'll have both N-terminal and C-terminal fusions to overcome that limitation.
Okay, second question is, what is the sensitivity of the assay?
Great question, and it's not, it's a complicated question because I can't just give a number. We do need to do some spike-ins with known affinity antibodies, and then for each of those, we could say, here's our limit of detection in terms of concentration. But certainly, we'll be more sensitive to higher affinity antibodies. You can also ask the question, if someone with a known or a group of individuals with a known infection, like HIV, and not, what's your sensitivity and specificity for detecting the infection? That is very high for most infections, especially if they're recent or chronic. But it does have lower sensitivity for historic vaccinations, like measles, is more difficult to detect in adults who are vaccinated in childhood.
Yeah, so I mean, like any serology, right? It's highly dependent on what you have in the serum. So have you thought about applying this platform to screening of menstrual blood for signs of infection or signatures of endometriosis?
No, but if someone is interested in that, please come visit us at the booth today.
Okay, next question from Joe. Hey, hi Joe. Can the antigen library include glycosylations, or is that a current limitation due to the use of E. coli?
Yeah, currently it's a limitation. Other PTMs we have put on, like citrullination, which is an important modification for rheumatoid arthritis antibody detection. But we have not done glycosylation, and I do think that using a eukaryotic translation would definitely help.
Next question is, how sensitive is IgE? compared to currently used clinical assays?
Yeah, so we're sort of in the early days of comparing clinical panels to our AIRR-C assay. And there's certainly a Venn diagram, and we're picking up a lot of things that the clinical assays don't pick up, and that's partly because they don't test for them. We're using a very comprehensive antigen library, about 2,000 proteins in the library. And there's some things that we're not picking up, potentially because they're post-translational modifications, non-protein components of a extract or something like that. Yeah, but we'll know more as we do more and more of this.
Yeah, or in some cases, I guess, really low levels of IgE, right?
IgE is a very low abundance molecule compared to IgG. That's true.
Okay, do you see differences between peptide and full-length anti-interferon antibody responses in COVID-19 with patients with different levels of disease severity?
Yeah, that's a good question. I think our N is too small to answer that now, but it's certainly something to think about.
Can you distinguish neutralizing versus non-neutralizing antibody responses?
Yes, with a caveat. So if we have done a prior study to associate, and if we're talking about like epitope libraries, peptide libraries, we have done a study in COVID-19 where we see which regions of spike or RBD correlate with neutralizing titers. If you build a model like that and then go and ask, oh, does this person have neutralizing antibodies based on their profile, you can make a prediction. But otherwise, I mean, you have to use correlation to answer the question. You can't tell just from the data unless you do some sort of modeling, which I think this community would be very good at, actually.
Yeah, that's a really cool idea. With respect to self-addition of barcodes on the proteins, how do you handle barcode collision problems?
I'm not sure what collision means, but if it has to do with difficulties in distinguishing related sequences, we're using sequences that are sampled from a huge space, so it's really not an issue, if that's the question. But if you need clarification, please come to the booth.
Yep. And how expensive is the assay? Is the cost practical for clinical use?
Certainly for clinical use, yeah. The question is going to be more about large numbers of research samples from academics with a budget. And again, I would urge you to come to the booth and chat with JP.
Yeah. Okay, I think we're getting close. Maybe we do one more question, and then if there are others that I didn't get to, please ask during the coffee break. So of the remaining questions, how about, do you know the affinity range captured in these assays? How do you exclude background noise? So sort of two different questions there.
Yeah,
for the price of one.
I think the ability to detect the antibody is a combination of its abundance and its affinity. Yeah. So hard to say with just one of those.
Right.
In terms of background, we on each plate of samples that we run will include mock IPs that include all reaction components except the antibodies. And so that's how we determine our background when we do our statistical comparison of each sample to the set of backgrounds.
Yeah, perfect. Okay, so I think we'll stop it there and go for coffee. Now, I want to emphasize we're going to coffee 5 minutes early. Please, 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 please be back here at exactly 11 o'clock, or preferably a little bit before. Thank you.